The polymerase makes a complementary copy of the template DNA, starting from each primer, and therefore copying the target region. At the end of this process, the total amount of target DNA will have doubled. Now we repeat the heating step, and the whole process occurs again. Once more, the number of copies of the DNA bracketed by the two primers doubles. Each cycle of this process results in a doubling of the target region. After 25 cycles of PCR, which means in less than 2 hours, we have 2 to the 25th power, about a 34 million-fold increase in the amount of our target DNA. In effect, the resulting solution, which started off as a mixture of template DNA, primers, DNA polymerase enzyme, and free A's, T's, G's, and C's, is a concentrated solution of the target DNA region. A major early problem with PCR is that DNA polymerase, the enzyme that does the work, is destroyed at 95 degrees centigrade. It was therefore necessary to add it afresh in each of the process's 25 cycles. Polymerase is expensive, and so it was soon apparent that PCR, for all its potential, would not be an economically practical tool if it involved literally burning huge quantities of the stuff. Happily, Mother Nature came to the rescue. Plenty of organisms live at temperatures much higher than the 37 degrees centigrade that is optimal for E. coli, the original source of the enzyme. And these creatures' proteins, including enzymes like DNA polymerase, have adapted over eons of natural selection to cope with serious heat. Today, PCR is typically performed using a form of DNA polymerase derived from Thermus aquaticus, a bacterium that lives in the hot springs of Yellowstone National Park. PCR quickly became a major workhorse of the Human Genome Project. The process is basically the same as that developed by Mullis, but it has been automated. No longer dependent on legions of bleary-eyed graduate students to effect the painstaking transfer of tiny quantities of fluid into plastic tubes, a state-of-the-art genome lab features robot-controlled production lines. PCR robots engaged in a project on the scale of sequencing the human genome inevitably churn through vast quantities of the heat-resistant polymerase enzyme. HGP scientists therefore especially resented the unnecessarily hefty royalties added to the cost of the enzyme by the owner of the PCR patent, the European industrial pharmaceutical giant Hoffmann-La Roche. The other workhorse was the DNA sequencing method itself. Again, the underlying chemistry was not new. The HGP used the same method worked out by Fred Sanger in the mid-70s. Innovation came as a matter of scale through the mechanization of sequencing. Sequencing automation was initially developed in Lee Hood's Caltech lab. As a high school quarterback in Montana, Hood led his team to successive state championships. He would carry the lesson of teamwork into his academic career. Peopled by an eclectic mixture of chemists, biologists, and engineers, Hood's lab became a leader in technological innovation. Automated sequencing was actually the brainchild of Lloyd Smith and Mike Hunkapiller. Then in Hood's lab, Hunkapiller approached Smith about a sequencing method using a different colored dye for each base type. In principle, the idea promised to make the Sanger process 4 times more efficient. Instead of 4 separate sets of sequencing reactions, each run in a separate gel lane, color coding would make it possible to do everything with a single set of reactions and run the result in a single gel lane. Smith was initially pessimistic, fearing the quantities of dye implied by the method would be too small to detect. But, being an expert in laser applications, he soon conceived a solution using special dyes that fluoresce under a laser. Following the standard Sanger method, a procession of DNA fragments would be created and sorted by the gel according to size. Each fragment would be tagged with a fluorescent dye corresponding to its chain terminating dideoxynucleotide. The color emitted by that fragment would thereby indicate the identity of that base. A laser would then scan across the bottom of the gel, activating the fluorescence, and an electric eye would be in place to detect the color being emitted by each piece of DNA. This information would be fed straight into a computer, obviating the excruciating data entry process that dogged manual sequencing. Hunkapiller left Hood's lab in 1983 to join a recently formed instrument manufacturer, Applied Biosystems, Inc., ABI. It was ABI that produced the first commercial Smith-Hunkapiller sequencing machine. Since then, the efficiency of the process has been enormously improved. Gels, unwieldy and slow, have been discarded and replaced with high-throughput capillary systems, thin tubes in which the DNA fragments are size-sorted very rapidly. Today, the latest generation of ABI's sequencing machines is phenomenally fast, some 1,000 times speedier than the prototype. With minimal human intervention, about 15 minutes every 24 hours, these machines can produce as much as 500,000 base pairs of sequence per day. It was ultimately this technology that made the Genome Project doable. While DNA sequencing strategies were being optimized during the first part of the Human Genome Project, the mapping phase forged ahead. The immediate goal was a rough outline of the entire genome that would guide us in determining where each block of eventual sequence was located. The genome had to be broken up into manageable chunks, and it would be those chunks that would be mapped. Initially, we pursued this objective using yeast artificial chromosomes, YACs, a means devised by Maynard Olson of importing large pieces of human DNA into yeast cells. Once implanted, YACs are replicated together with the normal yeast chromosomes. But attempts to load up to 1 million base pairs of human DNA into a single YAC exposed methodological problems. Segments, it was discovered, were getting shuffled, and since mapping is all about the order of genes along the chromosome, this shuffling of sequences was just about the worst thing that could happen. BACs, bacterial artificial chromosomes, developed by Pieter de Jong in Buffalo, came to the rescue. These are smaller, just 100,000 to 200,000 base pairs long, and much less prone to shuffling. For those attacking the human genome map head-on, groups in Boston, Iowa, Utah, and France, the critical first steps involved finding genetic markers, locations where the same stretch of DNA drawn from two different individuals differed by one or more base pairs. These sites of variation would serve as landmarks for orienting our efforts throughout the genome. In short order, the French effort under Daniel Cohen and Jean Weissenbach produced excellent maps at Geneton, a factory-like genomic research institute funded by the French Muscular Dystrophy Association. Like the Wellcome Trust across the English Channel, the French charity took up some of the slack created by insufficient government support. When in the final push, detailed physical mapping of BACs became necessary, John McPherson's program at the Genome Center at Washington University was the major contributor.